A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 7. Recent Advances in MS-based Proteomics. Lecture 1. Protein Quantification using LC-MS.
It is our privilege to have amongst us Dr. Debodip Bhattacharya. Dr. Debodip joins us from Thermo Fisher Scientific. Dr. Debodip Bhattacharya received his PhD in chemistry and biochemistry from Emory University, Atlanta. Dr. Bhattacharya is a senior manager of product marketing, triple quadruple MS for all businesses in the applied market division of Thermo Fisher Scientific. Dr. Bhattacharya has over more than 12 years of experience working with mass spectrometers and other high-end analytical instruments. He regularly engages with customers across varied industries and academic institutions. Further, he works closely with scientists, business development personnel, regulatory bodies, and key opinion leaders to address the needs of mass spectrometers, specifically triple quadruple users. With these words, we invite Dr. Debodip Bhattacharya from Thermo Fisher Scientific to deliver his lecture. Thank you.
All right, Dr. Dev, thank you very much for accepting. And despite a very hot time in Boston right now, you are giving a lecture at 12:30 late night, early morning. So we are very grateful that you're going to share your experience of triple quadruple based mass spec and target proteomics with the participants. So with these words, we are ready to hear your lecture, and please share the screen.
Thank you. Can you please confirm that you can see my screen all right?
Yes, it's coming well, and your voice is also audible. Please go ahead.
Fantastic. Thank you. Thank you so much, Dr. Srivastava, for this wonderful opportunity, and thanks everyone for tuning in. Must say that I'm probably not the first one to say this and not the last one, that these are unprecedented times and none of us were prepared to deliver something like this on our virtual platform. But yeah, man proposes and somebody else disposes. So without further ado, I would like to get started because time is of the essence. So to begin with, the focus of this presentation is around using mass spectrometry, or more so LC-MS technology, in the world of clinical implementation. So whether it is clinical proteomics, or it can be the clinical implementation in the hospitals, in the routine clinical market, in the clinical diagnostic labs, so on and so forth. So I'm going to do a little bit of a preamble about the market, so to speak, as to just highlight how this market is evolving. And I'm pretty sure in India, you are seeing this by and large becoming the norm, because it is moving from that kind of a day where you used to have, or we still have a lot of them, as the drugs that were given to treat a certain medical condition, a certain disease, and how it evolved in the present day world, where a lot more holistic information is being collected from the patients to better understand a patient. And by better understanding, from its proteogenomic, from their metabolomic profile, from their exosomal and endosomal situations, all of these things pulled together to essentially gives the physicians a much better idea as to how they can make or start a targeted, customized treatment regime for the patient in concern, which in turn offers much better chance of recovery, of treatment provided to the patient. Having said that, I'm not saying that the left side of the screen is not valid anymore. We still take our, paracetamols, we still take our acetaminophen and ibuprofens, and we still take a lot of the small molecule drugs, or in general, the drugs which are made for, by and large, for the entire population catering to a certain disease. Now, how we move from this generic to genetic, I mean, it's probably a tongue twister right over there, but how do we move from the generic to genetic pathway? So if I think about it, in the past, it used to be all based on symptoms, and there would be a lot of optimization of treatment regime, where in the present it is more focused on cohorts. And as I had the opportunity of listening to Dr. Suravanchi's presentation just before, right around the time when I joined, and how things are getting more importance about cohort studies. And it's all on the evidence-based medicine as to two siblings who born from the same parents and grew up together, but one they are probably poles apart in terms of their geographical location and how one is affected by a certain medical condition while the other person isn't. Their lifestyle, their exosomal conditions, that also has an impact on their metabolomic profile and their proteogenomic disposition, which in turn creates a lot of differences. Understanding that requires the transition of this implementation of the personal molecular profiles using non-molecular imaging technologies to other sort of waveforms, essentially something that refers to as all the scanning capabilities that we have, to compiling them into this big data analytics, where in a sense it's a library feature that is being developed, which creates these reference databases and creates these rules and algorithms and all of those aspects, where we are looking into the future, where certain conditions, it's almost similar to how you can go to a website, you can plug in certain information and it gives you right product, exactly what you are looking for. I mean, how you can refine your searches. It could be something that you're looking for a car or you are looking to buy a shirt, and how the websites are getting more and more streamlined to cater to your requirements based on what you're looking for. With all this data analytics and the databases coming in together, that's where the world is heading to, where there would be AI involved in, machine learning involved in, and where a certain condition, when added into the database, would give out the potential treatment regime that would work for the person with all his or her data available to us. So having said all this, there is a strong evolution in this particular world of protein therapeutics, protein-based treatments, which are becoming a norm. I mean, in today's world, if you dial into your TV, or every now and then when I see an advertisement of a certain therapeutics coming in, by all means, I mean, it's basically 99% of the time, the name of the drug has MAB at the end, which stands for monoclonal antibody. So we are seeing that there is a significant uptake in the number of monoclonal antibody or protein-based therapeutics that are coming out. And if you look into, typically FDA's approval pipeline, that's exactly how it's happening. Now, while this is happening, there is also an increased demand for essentially the LC-MS technology. And it can be for qualitative, which is quite well known. And as we saw in the first talk of the day, that was exactly highlighted, where high resolution accurate mass coming into play, providing better capability, better quality of results. However, the days of only relying on some sort of ligand binding assays, or ELISA or immunoassay-based technologies for quantitation, for determining, after you have determined how does this particular therapeutic look like and where does it go and bind to, The next question when it is applied, when it is given to a patient, the next question that comes into mind and would be asked by every physician is how much of the therapeutic is actually going into the body and staying in the body and making an impact, because based on that, they can decide on the dosage regime. So for that, quantitation is necessary. Now, if we track back 10, 15, 20 years, it's not that LCMS didn't exist back then, but the instruments probably were not to that extent, were not capable of delivering the levels of sensitivity, the levels of robustness that are capable in today's world. So if we think about it, there is an increased demand for LCMS because LCMS is also coming up and providing the necessary necessary weapons that are needed to cater to this particular world. So from selectivity, specificity, sensitivity, robustness, reproducibility, LCMSs are capable of doing that. And the reason how LCMS is managing to do it is because also in the last couple of decades or more than that, in the world of drug development in the bioanalytical phase between phase 1, 2, 3, LCMS has been extensively used. So that kind of demand actually catalyzed the process of bringing LCMS to where it is today. And today, based on what I just shared in the first couple of slides, when we think about this personalized medicine, the translational research, in other words, the precision medicine, so to speak, where biomarkers are being looked into, structural elucidation are super critical, understanding the holistic perspective from a proteomics, proteogenomics, also metabolomic data coming in, every condition's being taken into consideration. The implementation of LCMSs are becoming very, very important. So, needless to mention, understanding of this entire complex world is extremely important because it is not very simple to understand a biological system, especially when that system is not constant and consistent between one person to another. There's significant amount of change. I mean, how often do we get to see the situation where a whole family went out to have dinner somewhere and one of them ends up being sick and the other, family of 4, the other 3 is not even affected. So, or, in situations like COVID-19, you see that, 2 people who were in the same vicinity, one of them got it and the other one probably didn't. Or even if they did, both of them did, one was recovered or one recovered very fast while the other actually had to go to the hospital or, had to go through an excruciating process. So understanding the complexity of the system is like understanding what molecules are you looking into? What are your goals? What kind of molecules and what are the matrix or matrix interferences doing for you? The next part is choosing the platforms, because when it comes to understanding the problem, that's the first step. The second step is where you actually have to make a decision that should I actually choose this particular mass spec like a high resolution accurate mass, or is it a triple quadrupole? Or is it something else? And that is a major part where you figure out that, okay, now I know what I need to look for, but I need to more or less understand what's the concentration of this particular molecule in the system. So the automatic answer over there would be a triple quadrupole. And if it is not the case, and if you're trying to look for the proverbial needle in the haystack, then you are going into the high resolution accurate mass. Then comes the fun part where you are trying to optimize and develop your workflow. So remember, LC and MS are 2 boxes. They are connected together. What you do with it is a lot more important, because what's your sample prep protocol and how you take that data that is generated by your LCMS, how do you analyze that data and give a succinct, impactful, lucid information down the line to the hospitals, to the physicians, is going to determine the fate of the patient. So the workflows better be absolutely foolproof, well-tested, well-validated, robust, reliable, reproducible. And there comes the importance of ease of use. So from the platforms to the workflows to having confidence in the data. Now, I think, I'm preaching to the choir when I highlight the ability, the processes that are typically used when we are talking about large molecules in clinics. Because you guys work in the world of proteomics, and I don't necessarily need to elucidate or illustrate more on how you are working in the world of proteomics. But when we think about the clinical implementation, where you have been looking into thousands of proteins, and then you are narrowing down your list down to like 3, 5, 6 critical proteins, which could either be a marker or an endogenous biomarker, or it could be some sort of a profiling capability, which is exactly, based on some of the publications from Dr. Srivastava's group in the recent times. There are some specific markers that you can monitor to understand how a human system is going to respond at the onset of a certain viral infection. So coming into all those things, that there are the large molecules can be divided into the groups that you're seeing over here, whether it's large peptides or monoclonal antibodies, fusion proteins, therapeutic proteins having multiple domains, antibody drug conjugates, and then also oligonucleotides. So, and when you look into it, based on if you're trying to just monitor a certain therapeutic, then it's probably one approach. But when you are trying to monitor a human system to understand where the markers are in the endogenous, situation, which would be a certain biomarker would be a certain protein responding to a diagnosed as a certain biomarker would be upregulated, downregulated in presence of some sort of an infection. Now, like I said, that there were several decisions to be taken with your choice of mass specs. There are lots of decisions that you need to choose, or you need to take when you have to choose from your LC options. And then comes your sample preparations and further purification of your samples. So, and the fun just doesn't end over here, because when you are at the clinical level, you're trying to develop something that is robust. So for hundreds of samples that are coming in where you are trying to monitor IGF-1, or where you are trying to monitor the insulin, or looking into insulin that are generated by the body versus how much that was given to the patient who has been diagnosed as being diabetic. So understanding the endogenous insulin versus the exogenous insulin and all the derivatives of insulin that are present out there, all those things come into play when you have to deal with multiple charge states, which is a fun part of the proteins. They are not singly charged, unlike small molecules that we are typically used to. There is always an issue with a non-specific binding. So at the lab, we do our own ways of, adding rat plasma, adding, or denaturing the protein. Then there is the flow rate dependence, and that's where, your choice of LC becomes very important. So how is it that, certain flow rate would have an impact on a certain charge state or the intensity of the mass detected signal of a certain charge state? And then understanding the data processing capabilities. So what you were expecting based on your in silico digestion, and if you use something like Skyline or any other software to understand, okay, this is how the protein is supposed to cleave in presence of tyrosine, or in presence of trypsin, or in presence of lyc, whichever kind of enzyme that you think about. And then in reality, what you see is probably not identical all the time. It is great if it is, but it is not necessarily guaranteed. So some of the examples of, therapeutic monitoring of monoclonal antibodies, there are different ways one can look into, especially when you are working with high resolution accurate mass. And in the middle, you have the intact, which enables you to do the top-down approach. And then, you can break it down and do a more bottom-up approach with peptides by looking into the peptides. And also, there is limited cleavage where you can use something like, IDES or similar enzymes. There are multiple options where you are not breaking it down to as small peptides as you would for a bottom-up, but that's typically known as the middle-down sequencing. You are not necessarily always decoupling the sulfurs or, reducing the disulfide bonds, but you could do a middle down sequencing based on heavy chain, light chain combinations. So these are typically and widely known in the biopharma world when they are developing therapeutics, and high resolution accurate mass comes into play as a very strong player, as a partner in here. So from selectivity to mass stability, accuracy, to the ability to offer fast polarity switching. And when you gain on resolution, it doesn't mean that you have to compromise sensitivity. So you get high resolution, high sensitivity with minimal variance from one scan to another, gives the high resolution accurate mass the unique ability to cater to this world. So in particular, the Orbitrap technology that started back in 2005 has gone up in leaps and bounds. And, I'm pretty sure you have been exposed to a lot of it since last week. And, you are also seeing a lot of it, this week, and going to see a lot more. So Orbitrap technology has been working quite, in a non-stop environment, and there are constant improvements in that technology that has enabled from where Orbitrap technology was first used for proteomics peptide mixtures and where it demonstrated sub-ppm mass accuracy to the extent of in the recent times where a full proteogenomic analysis of a child or an infant patient was done or based on a focus on a specific disease, in this case leukemia, and also single cells are being looked into. Now, if I think about how do you use Orbitrap in the clinical world, come to think of it, there is this option of taking the full scan MS, and that always is the way to go for most of this high resolution accurate mass technology, because full scan always gives you a comprehensive picture of what's happening. And even if you miss out on something, you can go back and data mine and look into the data. There is targeted SIM, which can also be done post-acquisition. And then there is more targeted in form of parallel reaction monitoring that helps quite a bit in terms of doing targeted quantitation. So when we think about using high resolution accurate mass for targeted quantitation, the process actually is a lot similar to what you would do in a bottom-up approach when you are looking into a large protein molecule. So you can determine the peptide species, that would be your signature peptide, by using an in silico digestion capability. And you would select your peptides with your favorable properties for MS-based analysis. And then you can go from MS1 data to MSMS. And high resolution accurate mass are routinely used in this way, especially for proteomics, but also increasingly getting used in the world of routine clinical analysis. So I'll just show some of the examples over here. And this is something that we are using on our recently launched high resolution accurate mass with, which is Orbitrap Exploris 120. I'm not going to go into the product details, but just to give you an idea of all the isoforms that you can potentially see for an intact analysis of IGF-1, and you can compare it with, its internal standard, and you could do the full calibration curve, and also you can see that the robustness data is pretty good across, the entire panel of which are like 500 injections that were done. And, we have seen tremendous, lack of variance, which is always a good thing. So a high degree of correlation from one injection to another and throughout the calibration processes, as well as when we look into all the calibrator experiments that we are showing only 54, as I couldn't fit more data on this slide. So we are looking into all the calibrators, and you can see that, the robustness of the experiments is tremendous. And it doesn't vary beyond this level of, let's say, minus 0.5 to 1.5, which is perfectly acceptable across this 130-hour runs that were run during the process. So that goes to tell you a lot about how these research instruments are increasingly become amenable and usable in the domain of routine world. So then moving on to something which is like looking into the plasma proteomes, right? So there are lots of these things that are actually being looked into. So over here, we are using high resolution accurate mass. And in this particular slide, I'm showing quantitation of ApoA1, which chromatically elutes or co-elutes with some of the other high abundant proteins, making quantitation very difficult. So therein comes in the benefit of using a true high resolution accurate mass so that you can actually separate your signal and make it quantifiable. The fun actually becomes a little more when we go to some sort of thing like a medium abundance protein, transthyretin in this case, another plasma protein that is typically used to monitor certain level of infections, certain level of genetic diseases. And the same thing that can be monitored over here, that despite it being medium abundance and there is sufficient background interference, you can still quantify and isolate a signal. And so we didn't just stop there. We then looked into, so from high abundance to medium abundance to beta-actinin, which is a low abundant protein. And that's where you can still see that whether you are looking into a platelet-rich plasma or a properly prepared plasma after post-depletion of the platelets, we can still look into the specific proteins or peptides in this particular case and quantify them with a lot of confidence. So if I think about it, there is another major type of protein quantitation requirement in the clinical laboratories is looking into the quantitation of therapeutic monoclonal antibody, as I have been saying. And the application of immunotherapy is booming, and it is estimated that there will be almost like $100 billion in another couple of years or so. So there is a lot of potential over here of using this technology when I'm just talking about high resolution accurate mass in this world of looking into protein as an isolated or as a part of a bigger family of proteins, as part of proteomes, where you can actually have very succinct, well-resolved, confident data that you can look into. And here is one of those examples, particularly looking into COVID-19 samples. And there are plenty of publications that has come up that in the last couple of years. It's been there for decades now. I just highlighted some of the recent ones over here, looking into different ways of monitoring these things. And then, here's a portfolio of all the high resolution accurate mass instruments that we have. Depending on your use and choice, you can select from them. But then comes the other part that actually Dr. Srivastav introduced me with, which is focusing on, by and large, on the triple quadrupoles. So this is where it becomes a lot more routine, and perhaps understanding of what to expect is very critical. So once again, triple quadrupoles are the mass spectrometers that you would go to when you actually know what you are trying to detect and quantify them. So there are multiple ways you can do it, SRM or MRM, whichever way you want to call it, fragmentation followed by collision, followed by another layer of fragmentation and detection. And I already talked about PRM with the high resolution accurate mass. We also have something called high resolution SRM, which is extremely powerful in the world of protein quantitation. Now, what it does, it allows increased selectivity while increasing sensitivity, and I'll come to it in a couple of slides. So if I think about the triple quadrupoles for targeted quantitation of proteins, the process is very similar to what I described in a few slides back about high resolution accurate mass. So it's the peptide species that you need to understand how would your protein behave in the presence of a certain enzyme. Where do you expect the cleavages to come through? And so what would be your ideal signature peptide? And you start looking for them. But remember, now instead of the full scan, now you have mass information that you need to use for identifying and zooming in on specific mass fingerprints or fragments where you would only catch those particular signature peptides. So once again, You do not have to select with only MS1 data. You can go towards MSMS, as I described in the previous slide. And now what you are going to get is targeted quantitation of your critical peptides with a lot more sensitivity. So remember when I said that, few years back, a decade and a half ago, our mass spectrometers were not that sensitive, but now achieving high sensitivity is not really a big issue when you have a high-end triple quadrupole. And that's where you can actually now get similar sensitivity to what you would be getting with your immunoassays, but you have a lot more selectivity, a lot more specificity on your side, with which your confidence on your data is going to be a lot higher, because this is going by your mass fingerprint and it's not necessarily looking into similar structure that the same antibody or a certain antibody used for your immunoassay or immunocapture would attract any kind of peptides that shows that kind of functionality. Mass spec is a lot more specific and selective, hence gives a lot more robust and confident data. So these benefits, along with robustness, the capability of high resolution SRM, speed, high sensitivity, allows us to regularly use this for, especially in the world of protein quant. And here is an example that I would like to highlight, that when I said a couple of slides ago that HSRM or high resolution SRM helps tremendously. And here is a classic example. So we are looking at a particular peptide obtained from a digested monoclonal antibody hinge. And what we are looking at the bottom of the left part of the screen, we are looking into the internal standard. But when we inject the sample in the under normal conditions, you see the specific signal is quite low and is going to have a lot of matrix interference. So quantitation can be iffy at best. So now when you actually use the HSRM, where you can actually reduce the Q1 width of your quadrupole, which is very, very specific to the triple quadrupoles that I'm referring to here, that's the time when you are going to get fewer ions come through. So imagine holding a meeting in a room where you haven't closed the door completely, but it's ajar. So people who are not supposed to be in that meeting will not necessarily walk in. Only those who are supposed to be in that meeting would come in, knock on the door or open the door and come in. So yes, you might say that when you are reducing the Q1 width, aren't you going to have fewer ions of your interest as well? And absolutely. But you are reducing your matrix interference so much that that compensates for it, as is the case over here. So sensitivity is typically defined as signal to noise ratio. So while the area under the curve goes down when we go from a fully open first quadrupole to 0.2, which is, very narrow first quadrupole, the area under the curve goes down by a factor of 4. However, the signal to noise ratio goes up by a factor of 4. So you actually get much higher sensitivity. And not just that, here is an example of monoclonal antibody digest, another one, where we are looking into the heavy peptide and the light peptide. Now you can see the light peptides don't necessarily get affected too much in this process, but between changing from a fully open Q1 to narrowly open Q1, there's a lot of the matrix interferences that you have gotten rid of for the heavy peptide, which gives you much better quantitation capabilities. And as we can see over here, here is an example of a combination of C-peptide, parathyroid hormone, and insulin. And it's a 6-minute runtime where we are quantifying all of them. So high speed, high sensitivity leveraged by the HSRM. This is equivalent to the workflow from where we took a high resolution accurate mass of looking into plasma proteins and converted that to a triple quad. This is one of those examples that we have recently reported. It's a longish because we are looking at much higher number of proteins. We are looking into also the TDM, or therapeutic drug monitoring of the tumor necrosis factor, TNF alpha inhibitors, and how using certain drugs or certain therapeutics like adalimumab, infliximab, these are all biotherapeutics that are available in the market today, and how the anti-drug antibodies are generated in every patient, which varies between one patient to another, and just monitoring them on a triple quadruple and quantifying the adalimumab or infliximab signal on its own, and the ADAs or anti-drug antibodies made by the body to respond, to stop, or immunogenic response to the drug, how one can determine the efficacy of the drug and hence determine the treatment regime. And this is looking at a much bigger cohort of proteins, where we are looking into, like, close to 500 proteins and monitoring them across the entire whole blood cell. So here is, a detailed explanation of what we are trying to do. So remember, when it comes to C-peptide, you have always, when it comes to these proteins and peptides, on a triple quadruple platform, because the speed is so high, you don't necessarily compromise on the data quality or on your confidence. So you can have close to 660 SRMs per second. I still remember when I was in my graduate school, my PhD advisor told me that, when you get a 2 millisecond dwell time on the mass specs, Star Trek would be real. And Star Trek is still not real. It's still in the movies. But these are operating at 1.5 millisecond dwell time. So that's a remarkable achievement, which also allows the ability to do faster collection within the same time frame, 6 minutes, but you are actually monitoring multiple transitions to be absolutely sure that you are truly monitoring the C-peptide or the parathyroid hormone or the insulin in question. So there are multiple ways you can actually confirm the data. I already talked about the quantitation of, free adalimumab and infliximab, and in the interest of time, I just, skip over to the next part, which is perhaps a lot more interesting and pertinent under the present circumstances, where we are looking into the COVID-19 spectrum. So if you think about it, the COVID-19 spectrum, there are multiple ways that are being looked into. One is, of course, there's the option of virus research, where people are looking into the viral proteins and the viral proteomes and understanding the virus, and also monitoring options to monitor the virus. And then there are testing of the patient, understanding how much viral load they have, and also looking into the patient populations or the patient in concern health conditions by monitoring certain key markers in their plasma, which is exactly what Dr. Srivastava's recent publication focuses on. And then there is the vaccine research, which focuses on understanding what needs to be done and what should be the approach. Should it be a messenger RNA approach to block the translational process of the virus once it is inside the body? Or is it just like, a viral load, a payload to be incorporated or injected in the human body that will counteract the entire virus? So there are different approaches that are out there and, are being converted into the vaccines. So what we are looking into, this is a publication from the Mayo Clinic, where what they have done is looked into the entire perspective in a high throughput environment by using high resolution accurate mass. And they have taken hundreds of nasopharyngeal residual swab samples, monitored it with, first detected positive with RT-PCR, and then they are looking at it by monitoring certain typical critical identified nucleocapsid proteins. And they are using additionally FAMES for increased selectivity on the mass spectrometer to get increased confidence in data, in other words. And when I move on to the other one, this is a triple quadruple technology that is being used by this group or the organization called Grupo Fleury, based out of Brazil. And as luck would have it, they're actually using it on the patients right now. So almost 25% of the daily samples that they're analyzing by RT-PCR are being sent to LCMS where they are not 100% confident that if this person is COVID-19 positive or has some sort of other viral infection. So that's where LCMS is coming very handy, because it's looking at it from a granular perspective by monitoring specific peptides that come from the viral mRNA or the nucleocapsid proteins of the virus, and not necessarily something that would be unanimous or universal for any viral infection, whether bloodborne or airborne pathogens. Another thing that is becoming very important is use of paper spray technology, which is being used in conjunction with the mass spectrometer or the triple quadrupole over here. And this is coming out of the Department of Chemistry and University of North Texas, where they are using this whole paper spray technology with a certain matrix where they spot the sample, or they are actually taking the breath samples from patients. It's not nasopharyngeal swab, it's not oral fluid, it's not plasma, it's not blood. They're taking the breath analysis in a device, and you can see the bottom of the screen how the picture is, and then it goes and gets the effluent coming out of the breath samples gets captured on a certain specific paper, which they have registered as Teslin. And then VeriSpray or paper spray technology is actually a probe that looks into or that ionizes the sample on the paper itself and transfers it to mass spec. So no LCs involved in here, directly transferring the sample over to mass spec. And then I don't probably need to do much over here because, I'm preaching to the choir if I have to spend too much time, but this is a wonderful, wonderful piece of work coming out of Dr. Srivastava's lab, where not just the extent of study, and it is not a Me Too data that I would like to highlight. It's groundbreaking as to how you can monitor a patient ahead of time. Early onset of infection, peak infection, and post-recovery, and looking at the typical plasma-oriented markers that can give the physician a very good idea of where the person is with regard to being infected or not. So it's a holistic pathway, and that is such a big, powerful capability to the entire entire hospital community, entire physician community, to know that there is another way other than monitoring just the viral load or just the virus, but to look at also from the patient's perspective. So globally, a lot of these things that are happening around, I don't want to spend too much of time, as I want to just focus on some of the critical aspects pertaining to the software that can enable peptide quantitation with regard to triple quadrupole. So, there is a research of global omics and distribution amongst the patients. So looking into thousands of proteins and then narrowing down. Then there is the importance of looking at the therapeutic regime on certain specific monoclonal antibody-based therapeutics, such as the one from Eli Lilly that has been approved by US FDA, that are being used on COVID-19 positive patients for treating them. And then there are certain other aspects, such as looking into the identification and monitoring specific markers in the nucleocapsid proteins or the viral load that helps in understanding the situation that the patient in particular is actually in right now. So without further ado, I'm almost down to the last section of my presentation over here. I just want to focus a little bit on the only thing that you would use to communicate to the mass spectrometer. I mean, as we would all wish that we would like to see the detector and we would like to see the quadrupoles, we would like to see the source, those things are actually inside the box and you don't get to see. The way we speak to the mass spec and to the LCs through the software. And that's where the importance of software becomes extremely, extremely important. I cannot emphasize how many times I have seen from my days in the lab to my present time, where people are actually asking more and more questions about software, where I'm getting constantly, asked questions about how to make the software even more user-friendly. So this is where with the TraceFinder software, where peptide quantitation can actually become a lot easier going forward for each of you present, who are watching this presentation with an inordinate amount of patience. So as you know, you're looking into the entire in silico digestion, and then there are certain things like the Predictor tool, which are present in the trace finder that can create a database of masses or transitions to quantify. And you don't necessarily need to be a software expert to have that. And this, in turn, over a period of time, creates a library. So a lot of the times when you are injecting similar peptides, let's say from insulin, you go to a liraglutide or some other form of glutides, the cleavage pattern would be somewhat similar, and the software automatically will guide you through. So ideally speaking, you will have the choice of setting your digestion. Is there a question? Okay. So ideally speaking, there is an option for you to set your digestion settings, and you can choose your enzyme. You can set your transition settings, and you can choose your, your Y ions and your B ions, and it generates your predicted peptide list. And also you can actually incorporate all the modifications that you think that there would be. And you can also import protein or enter peptides and select the digestion rules, as I just said, transition or isotope criteria to get the mass list, and can also add specific modifications as one would expect. Then you can also select or name your database to save the predicted peptide masses in the target peaks or targets with confirmation of fragments. So that's a big advantage as you are just beginning to create your own library of peptides, which you can leverage and capitalize on as the day goes by, as you get more and more analysis done. And then you have the mapper tool, where now you can do any of your in silico digestion. You can import any of the CSV file from any application to a certain compound database, and that can do the automatic mapping. And you can use it in this particular way. You can take any CSV file and map it with the drop-down categories, and what you found in your in silico digestion would also map out with what you are seeing in real life. And then that makes your life a lot easier, because what you identified via the in silico digestion as your signature peptide becomes almost, if you see the same thing, which you should be able to, hopefully, fingers crossed, once you see them, you can see that, okay, if there are no differences between the predicted RT and, what you're finding, the precursors, the product, the collision energy, and so on and so forth, and you have your identification done. And in addition, these platforms also offer a lot of other things, like, you can, it gives you an automatic suggestion based on your molecule of interest as to what would be the desirable parameters when you're setting up a method. If you have an existing method that you want to convert, yes, that can also be done. And the software automatically, there's an AI part of it that will automatically suggest you that these are your likely parameters based on your data that you would share. And then there is the compound optimization tool, which would help you in optimization of all your SRM parameters, whether you are using via direct infusion or if you're using an autosampler. Last but not the least, I would say that, it is capable. These mass spectrometers, today's mass spectrometers in general, triple quadruples, would be able to take on any of your LC inlets, from high sample throughput, multi-channel systems to nano LCs. And, as I'm sure you've been exposed to the data, I don't want to spend too much time on the description of the products. There are primarily 3 triple quadruples that we talk about at Thermo Fisher. The entry level is the TSQ Fortis, the mid range is Quantis, and the high level is, or high end is the Altis. Almost 100% of the data that I shared today on the triple quadruples were obtained on the Altis, because of, you get HSRM facility, you get highest sensitivity, which is so much required, especially when you are working with, background interferences, which are also peptides. So you're looking for the needle in the needle stack. So in summary, as I said, LCMS is actually gaining quite a bit of popularity, and as much as one wouldn't want to ever live the situation that we are going through as humanity in today's world, COVID-19 actually, I think, has taught us a lot of lessons, especially from a diagnostic perspective, as to the importance of having complementary, orthogonal, holistic evaluation perspective of monitoring patients, of detecting patients who are positive, and once detected positive, of monitoring them. And LCMS is, because of its inherent specificity, selectivity, especially more so for the triple quadruples, they are getting increased importance in that world. High resolution accurate mass offers a lot of advantages, and it gives you both characterization, qualitative and quantitative capability. But when you are trying to actually quantify a certain particular peptide, whether it's an endogenous biomarker or whether it's a therapeutic that you're trying to monitor and quantify for a pharmacokinetic, pharmacodynamic perspective, or if you're looking into standard TDM or therapeutic drug monitoring capability in the clinics, in everywhere from where you need sensitivity, you need the instruments to be supremely robust. You need to have reliability. You need to have reproducibility on your from your instruments and in your workflow. And that's where triple quadruples are the most reliable and sensitive and robust instruments that you can actually bank on. So having said that, I'm at the end of my presentation. Before I hand it over back, my sincere gratitude to Dr. Srivastava and everyone at IITB. It's my alma mater. I mean, that's where I got my master's from. So it's always pleasure to present in front of an IIT Mumbai audience. And my association with Dr. Srivastava is not really very new. It's been for a few years, and it's getting stronger with every passing day, which I'm loving to bits. And my sincere gratitude to every one of you who have been listening to me with deft patience and attention. Thank you so much.
Great, wonderful lecture, Dr. Dev. Thank you so much for sharing your experience of so many years working in this area. And it was very much in the flow of exactly what we have been in this training program. Yesterday, we talked about targeted proteomics workflow, and we discussed with the participants about running sample in the artist and doing the quantitation. And then we had sessions on panorama. And then we had Dr. Shubhra, who also talked about different validation strategies. So overall, I think, what I'm sure all the participants are getting. In fact, Dr. Mark Baker, he spoke about colorectal cancer biomarker and their validation with the targeted approaches. So I'm sure participants are appreciating the way things are coming. I think, mass spectrometers have really become very important for even validation and successful clinical translation of various biomarker and the different proteins of interest. And you showed some of the interesting examples of COVID-19. And I must say that in this time, there was literally no possibility of looking for any other reagent, right? Like we had no time at all to find out the reagent for, doing any antibody or some other validation. So whatever, if you have mass spectrometers, you can try to develop new assays on it. And that's where I think you showed that different groups from the world, they all tried innovative methods of, viral detection and prognosis part. So with this, I think, we are very thankful that you accepted the invitation and delivered this lecture, especially such a late night from Boston in the US. It must be very cold and snowy over there right now. And, despite all the odds, you accepted and delivered the lecture. Thank you so much. I'll open up for one or two quick questions from the participants. So Meher, please go ahead and ask a question. Meher, if you can unmute yourself, yeah, go ahead. You're not audible. Anyone else want to have any question in the chat box? Please write your questions for Dr. Dev or for Dr. Amol so that we can take the question. So in the meantime, while people are trying to get connected, Dave, I think what you showed was the remarkable advancements in the targeted validation strategies, and especially a lot of duty cycle which has changed, and how the assays can be much robust now, much faster, which was not thought of, right? How you see, like maybe in a very quick nutshell, if you see, like, with your extensive experience in the field, how you see the field is going to progress and make a path for the clinical translation where clinicians will not have hesitation of using a mass spec. Are there some plans when there are some more handheld or maybe bench top kind of very simple mass specs are being developed, where we are actually heading right now in this area?
Yes, I mean, this is always in the world of, and not just in the world of clinical. I mean, it's also in the food safety, environmental safety, where people are looking for portable instruments that they can take directly to the source, whether it's looking into environmental toxins or looking into pesticides or looking into this world, like, where you can roll in a mass spec, which in today's world is not. So we are not there yet as of now, but I would say that at this point of time, I expect mass spec to become synonymous to how MRIs are in today's world. It is a complex technology, as is the case with magnetic resonance imaging, but I think it has crossed that barrier, and I think that's where LCMS is now heading towards.
Absolutely right. Yeah. So participants, I think Ankit or Meher, you want to go ahead? Please ask.
Yes, sir. So Meher here from Ankit's laptop. So because I could not speak from that system. So, sir, my question is related to the data acquisition using MRM on Altis, triple quadruple Altis. So, sir, for the last few days, we were doing some experiment for MRM and PRM, and we saw that mostly because MRM is used for the validation of proteomics data. So we observed that most of the peptides that came in the discovery data in Orbitrap fusion kind of instrument, they are not detected or in the Altis, but and sometime we have doubted our sample, that sample is not good or something. But then we tried it to detect using PRM in the same Orbitrap instrument. So very good peptide with a good peak shape as well as intensity were observed. So I am asking you, what could be the possible reason for this? At least for the same peptide, if we are monitoring all the peptides, so one or none peptide is seen in MRM, but many peptides are seen in PRM. So, what are your views on it?
So of course, Meher, I mean, it's a very good question, and it's also a common problem because typically triple quadruples, the way fragmentation happens in the quadruples is very different from PRM. PRM is basically, it's still something that is leveraging on the full scan capability, and it's a different physics, so to speak. So without going into the details, there can be more ion suppression happening, which might be masking some of your peptides that you are seeing. So in other ways, I would say that, we can look into, if you want to email me, on this, or offline, Meher, and I can talk to you about, looking into different mass windows that we can potentially look into on the triple quadrupole and see if that helps identification and monitoring of these peptide signals, as opposed to as what you are seeing on the PRM.
Right.
Okay,
I think
sir.
it
Thank
would
you.
also be here, case-specific things, which is subject to collision energy and other optimization as well. But yeah, please do share the specific issues which we can take forward later on.
Okay, sir.
We are
Sir, one
getting
more question.
slightly late, Meher, so I have to stop you and other participants as well, because the next speaker is already ready, and we are actually running into kind of overlapping the sessions now. And participant unfortunately has no break, actually, even not even tea break we can provide them. So we are online has become probably much more hectic for all the participants. But we really appreciate they're all there, and we are very thankful to the speaker of the first session. So let me thank Dr. Amol and Dr. Dev once again for delivering the wonderful lectures from the organizing team IT Bombay side here.